Now let's discuss white blood corpuscles, which we also call as leukocytes. Well, leukocytes are termed as white blood corpuscles because they are colorless. Why they are colorless? Because as compared to red blood corpuscles, they do not contain hemoglobin. So one very important, you can say, feature of this is that they contain no hemoglobin. And when we discuss about the number of white blood corpuscles and the leukocytes, you can say they are around 6,000 to 8,000 per millimeter cube of the blood. So this is the total leukocyte count. So it varies from person to person. So as compared to red blood corpuscles, the leukocyte count is quite less, as you can see. So if we compare, if we want to give the ratio of WBCs is to RBCs, it is approximately 1:700. So this is the ratio. The RBCs are much larger in number, the most abundant cells of a body, as compared to WBCs, which are quite less in number. They are rounded or have irregular shape, and they can change their shape like amoeba. So they can show this amoeboid movement also. And because of this shape, they can actually squeeze out from the blood capillaries, and this phenomena is known as diapedesis, right? Because of this shape and the movement shown by them, they can squeeze out from the blood capillaries, and this process is known as diapedesis. And the size is fairly large as compared to red blood corpuscles. It is 12 to 20 micrometer. So as compared to RBCs, they are much larger in size. Since they are colorless, they do not contain hemoglobin. When we talk about their structure, then they have a cell membrane, there is a nucleus. The red blood corpuscles, they were anucleated, or they were without nucleus. But white blood corpuscles, they are nucleated, they have cytoplasm, and the cytoplasm contains these organelles, that's mitochondria, Golgi apparatus, centrioles and other organelles, right? Now let's talk about the type of white blood corpuscles. They are basically differentiated into two types. We have agranulocytes and granulocytes. Agranulocytes, they actually do not contain granules in the cytoplasm, whereas the granulocytes, they contain granules in the cytoplasm. So let's first of all discuss the agranulocytes. Agranulocytes, they are of two types, lymphocytes and monocytes. Lymphocytes form 20-25% of these leukocytes, and monocytes form 2 to 10% of leukocytes. So in lymphocytes, they are smaller in size, whereas the monocytes, they are larger in size. As you can see the image of a lymphocyte, it has a small size, scanty cytoplasm is there, and a very large round nucleus, right? And it is non-motile and non-phagocytic. So if we compare lymphocytes with monocytes, look at this image of monocytes. They are larger in size, they have much cytoplasm, and the nucleus is not round, it is bean shaped. So monocytes, they have a bean shaped nucleus, and they are motile and they are phagocytic. They engulf bacteria and cellular debris. And these monocytes actually turn into macrophages when they actually come in contact with the tissues of your body. So I hope that agranulocytes, they are clear to you. We have lymphocytes and monocytes. Then talking about granulocytes, they have an irregular nucleus which is lobed. They have granules in the cytoplasm and a nucleus which is irregular, or you can say it is lobed. Based on the staining properties, granulocytes are divided into eosinophils, basophils and neutrophils. Let's see first of all the eosinophils. Now this eosinophils, as you can see in the image, they are two-lobed. The nucleus is what? The nucleus is two-lobed, and you can see the granules in the cytoplasm. So they are two-lobed, they have coarse granules, and they take acidic stain, that is eosin. They take the acidic stain, that is eosin, hence their name is eosinophils. And they help in dissolving blood clot. They are non-phagocytic, right? And they also secrete lysosomal enzymes to actually kill the bacteria. Then moving on to basophils. These basophils, they have three-lobed nucleus. The nucleus is, as you can see, it is three-lobed. They take the basic stain. That basic stain is methylene blue. That is why the name is basophils. Like they take which stain? Basic stain, that is methylene blue. And they have less granules. And they secrete heparin, serotonin and histamine. So this is also one of the main feature of basophils. Then we have neutrophils. Now neutrophils, as the name suggests, they are neutral. They take both type of stains, acidic stains also and basic stains also. They have many lobes. As you can see in the image that neutrophils, the nucleus of the neutrophils, they have many lobes and they have very fine granules, and they possess small spherical lobe attached to nucleus by stalk lobe dense like structure which we call as drumstick. In mammalian females there is a structure in the neutrophils which we call as drumstick or bar body. This bar body is termed so because it was discovered by the scientist whose name was Bohr, right? So this is formed because of the transformation of X chromosomes. This structure, this drumstick or the bar body neutrophils is formed because of the transformation of the X chromosomes in the mammalian females. And these neutrophils, they actually eat harmful germs, so they are phagocytic in nature. Now if we talk about the number, then we have the least number of the basophils, we have the highest number of the neutrophils, and then we have eosinophils. So the most number, the larger ratio is of neutrophils when we talk about leukocytes as a whole, right? So now here I want to introduce to you a term that is leukocytosis. We have leukocytosis. When there is abnormal increase in the number of white blood corpuscles, we call the condition as leukocytosis, right? And then if there is an abnormal decrease in the number of leukocytes, we call that condition as leukopenia. So abnormal increase in the number of WBCs is termed as leukocytosis, and abnormal decrease in the number of leukocytes is known as leukopenia. Now where these leukocytes are formed, the formation of the leukocytes is termed as leukopoiesis or leukocytosis. Granulocytes and monocytes, these all granulocytes, that is eosinophils, basophils, neutrophils, and from agranulocytes, monocytes, they are formed in the bone marrow. They are formed where? They are formed in the bone marrow. While lymphocytes, they are formed in the lymph nodes, spleen, thymus, tonsil, bone marrow, and Peyer's patches. These Peyer's patches are there in the small intestine. These Peyer's patches are actually the clusters of the lymphoid tissue in the small intestine. So here the lymphocytes are formed. Remember that these four, monocytes, eosinophils, basophils, neutrophils, they are formed in the bone marrow, while these lymphocytes, they can form the lymph nodes, spleen, thymus, tonsils, bone marrow, and Peyer's patches. Then the lifespan of granulocytes, it's around 4 to 8 hours in the blood, while 4 to 5 days in the tissues. And if we talk about lymphocytes, then the time period, the lifespan actually varies from few days to a few months or year in fact. And the monocytes, they survive for 10 to 20 hours. So when the WBC count is actually increasing, this means you are suffering from some infection, maybe a bacterial infection or a viral infection. So WBC count is actually helpful in diagnosis of the disease such as typhoid, tuberculosis. So remember that the main important points regarding WBCs is that they do not contain hemoglobin and they have a nucleus and they show amoeboid movement, and this helps in the squeezing out of the leukocytes from the blood capillaries, which we call as what? This process is known as diapedesis. Then there are two types, agranulocytes and granulocytes. So agranulocytes, we have lymphocytes and monocytes. They do not have granules in the cytoplasm, and in granulocytes we have three types based on their staining properties, that is eosinophils, basophils, and neutrophils. Out of these three, neutrophils are the most abundant cells, 60 to 65%. However, the least number of cells is of basophils, right? Only 0 to 1%. And they are stained accordingly. Eosinophils, acidic stain, that is eosin. Basophils, basic stain, that is methylene blue. And neutrophils, they are neutral, they can be stained by both the dyes, acidic also and basic also. So I hope that white blood corpuscles, leukocytes are actually now clear to you.